All right, so as a review of what we did in lab this week, we were comparing or converting the transmittance of hemoglobin into the percent oxygen that was saturated inside that hemoglobin. So to do this, we were taking a spectrophotometer and then a vial of hemoglobin. Right here. So we were basically measuring the amount of light that passes through this hemoglobin. So the transmittance of this hemoglobin at a certain wavelength, 606 nanometers. So we were measuring that transmittance at different pressures. So we were modifying the pressure inside of that flask using a vacuum pump. And so we were changing the pressure, decreasing the pressure inside that volumetric flask. And then at the same time, we were dropping down partial pressure for the oxygen inside of that flask. So partial pressure is really just a proportion of oxygen or whatever target gas we want to consider. It's really just a proportion of that gas within a volume that we're comparing. So to do this experiment, we made a couple assumptions. The first was that at 100% oxygen saturation, we had a certain transmittance value. I think the average for all classes was somewhere around 60% transmittance. And so we were assuming that at our sea level, at the baseline that we were comparing, 760 millimeters of mercury, that we had 100% oxygen saturation. So there was a fairly high partial pressure of 160, or roughly 159.6 millimeters of mercury for oxygen. So that was one of the assumptions that we made, that at sea level, we have 100% oxygen saturation. We made a second assumption that at our true vacuum, where there's no oxygen left in that volumetric flask, that there would be no oxygen left for that hemoglobin. And so at 0% oxygen saturation, at 0% or zero partial pressure, we would have 0% oxygen saturation. So we took the transmittance of several values. I believe the average is something like 25. So since we have 2 points, we can draw a line between the two of them, like that. And then we can start making inferences from the data. So what we did is we took the transmittance values for every single one of our intervals that we were measuring. We plotted it along this line in order to determine the percent oxygen saturation that corresponds to that percent transmittance of that hemoglobin at a certain pressure. So from there, we had values for percent transmittance. We calculated the partial pressure of oxygen within each of these vials. From there, we made an oxygen saturation curve. So once again, we have partial pressure on the x-axis. You can't see the x-axis. We have partial pressure on this x-axis right here. And so this is the portion of that volume inside that sidearm flask that is oxygen. So if we consider an average atmosphere, there's roughly 20.9%, it's made of roughly 20.9% oxygen. We rounded that up to 21%. And then we calculated that by multiplying 0.21 times 760 millimeters of mercury to get 159.6 millimeters of mercury as the partial pressure at sea level for oxygen. So we then compared that to the oxygen saturation that we inferred from the previous graph, where at sea level, our hemoglobin is 100% saturated with oxygen. So we put a point here, and then we do this for every single one of our intervals that we made. As you can see, I added a bunch of them, just so you can see what a oxygen saturation curve really looks like. If I was smart, I would have saved all your data and we would have plotted all that together. So that way we could have had a empirical distribution of a oxygen saturation curve, but I didn't plan it that far ahead. So instead, we'll just use the one from my made-up data. So we have partial pressure on the x-axis, we have oxygen saturation on the y-axis. when we connect these dots, just drawing a curved line, we get a linear or a oxygen saturation curve that is what you would see in a natural environment. So you can see right here, you have 100% oxygen saturation. I don't know if you can see my writing on the board. Let me use a darker pen. We have 100% oxygen saturation up here. Over here, we have 0% oxygen saturation. So this is what's happening inside of your body. Every time you take a breath, every second that your blood's pumping from your lungs to the capillary beds, to tissues that need that oxygen the most. So in your body, your lungs are about as high a partial pressure as you can get. It's not exactly going to match 160 millimeters of mercury. It's more like 100 millimeters of mercury partial pressure for oxygen inside of your lungs. So if you think about it, your lungs are not entirely efficient. For every breath you take, that's weird. For every breath you take, you're only replacing, I think it's like a third of the air inside of your lungs. So your lungs are not going to be extremely efficient, but they have a much lower partial pressure than the outside environment does at sea level. So right here, we have about 100% oxygen saturation for our hemoglobin still. But once we get past this point, this line right here, which is basically where your lungs are, so your lungs are right here, the affinity for, or your hemoglobin's affinity for oxygen drops off fairly steadily. So as soon as your body starts pumping that oxygenated blood around the body, it starts losing oxygen fairly quickly. So once it loses, I believe, like 2 oxygen atoms, those heme groups just release oxygen much faster, which causes this very sharp decrease that you see closer to 0 millimeters of mercury. So this is an oxygen saturation curve. If you want to divide this up even more, you could put sections here. So you can have, I believe, veins on one line. So veins, oops, I can spell veins right. Veins are going to be pumping deoxygenated blood, or blood that's already passed oxygen onto cells and is now carrying carbon dioxide back to the lungs. So these are going to be extremely low in oxygen. You'll have your arteries. I can spell arteries right. Right there. So these arteries are highly oxygenated in blood. Those are carrying oxygen to whatever cells, tissues, organs that need oxygen. Everything else, so veins are losing oxygen really quickly. Arteries are carrying oxygen to whatever cells need them most. Yeah, so that's an oxygen saturation curve extremely quickly. I love, but I give the example of Olympic track runners modifying this partial pressure curve to suit their athletic capabilities. So Team USA's Olympic track runners train in Colorado Springs in Colorado, where it's roughly 6,000 feet in elevation. So they have a much lower partial pressure of oxygen available in that atmosphere for their bodies. So while they're training, while they're running, their body's going to be demanding a large amount of oxygen, but it can't fulfill that need because there's not very much oxygen in that atmosphere available for them. So their body's going to be trained basically to do more with less. So their hemoglobin will reach a point where it can be maximally saturated with oxygen, even though they have a much lower partial pressure in the air around them. So this is why, like I said, those track runners, they'll travel to Colorado Springs. They'll have a much higher, or their hemoglobin will have a much higher affinity for oxygen, so that they can do much more. They can run faster, jump higher, longer, whatever, with less oxygen, which makes them the athletes they can be. So this partial pressure curve can be modified by pH. So as we're respiring, as our cells are taking in oxygen, metabolizing, and then producing carbon dioxide as a waste product, the pH of the blood is actually increasing, so it's becoming, decreasing, so it's becoming more acidic. So if you wanted to draw another line on here, I'll use red. If you wanted to draw another line on our oxygen saturation curve for a higher pH blood, or more acidic blood, it would look something kind of like this. So it just falls underneath that oxygen saturation curve that you see in normal pH blood. So I believe a normal pH is something like 7.4 maybe. More acidic blood is going to have a much lower affinity for oxygen. So it's not going to be picking up oxygen, it's going to be picking up CO2 instead, and then carrying that CO2 over to the lungs where it can be exhaled. And then the whole process repeats itself. We breathe in again. That oxygen is passed from our lungs down, or travels through the bronchi, to the alveoli, through the capillary beds, into the blood, where it travels throughout the body, gets delivered to the cells that need it. Those cells produce carbon dioxide, and the blood becomes much more acidic. It loses its affinity for oxygen. It picks up CO2 instead, and then that blood travels through the veins back to the lungs where it's exhaled from our bodies. So yeah, that was the point of this lab, so that you would be able to see an oxygen saturation curve. I don't know how well you can see it on this video, but I'll be sure to post a couple of examples on Blackboard. So we were using hemoglobin, which was kindly provided by Mark Clausen. He's one of the 3 people who probably make this department run. So shout out to Mark Clausen and his lab. The other respiratory pigment that we don't have access to, but that is fairly common, is hemocyanin. So hemocyanin is found in merostomata, crustaceans. So horseshoe crabs, regular crabs, crustaceans, stuff like that. They'll have hemocyanin, and that will be a light blue color because the heme groups actually have copper instead of iron. So in hemoglobin, it gets this really red color because of the iron inside of that hemoglobin. So the actual hemoglobin molecule itself is basically colorless, but the heme groups attached to that hemoglobin molecule are red in color. So this is the respiratory pigment that every single chordate vertebrate will have, and many invertebrates also have the same respiratory pigment, just because it's fairly efficient for what it does. There are a bunch of other respiratory pigments that are fairly minor. There's Hemo, I don't know, I can't remember the names, but they're not found in the quantities found. It's like hemoglobin or hemocyanin. When you get to much smaller organisms, they won't have hemoglobin or hemocyanin. They just rely upon diffusion of oxygen into their bodies through cutaneous respiration, and then the diffusion of CO2 outside their bodies through the same process. So that's a limitation on size for those organisms. They can only draw in so much oxygen to a certain depth of their bodies before they start asphyxiating cells. So those cells will run out of oxygen if it grows too big. So that is one of the reasons why I love to keep harping on the surface area to volume ratio. So the larger surface area you have and the smaller volume that's inside your body, there's a necessity for more complex respiratory structures like what we have, lungs, or the exposed gills and axolotls. What else? Yeah, I'm sure you could come up with tons of examples, but yeah, so I think that was basically all we covered this week, was respiratory pigments, oxygen saturation curve. Yeah, so next week is Echinodermata, or actually several phyla. So we're going over basically sea stars, a little bit into hemichordates, chordates, and then Urochordata. So we have 4 different phyla to cover in a single hour-long class. It's going to be a very long lecture video. I apologize in advance. I know some of you in that feedback that I was asking for in the quizzes were asking for shorter videos. I'll probably break it up into 2 or 3 videos they can watch at your own pace over the course of those 3 days before lab when they're posted. So yeah, that should be about it. If you have any questions, shoot me an email or just raise your hand next class and I'll be sure to answer it as best I can. See you Wednesday or Thursday.